Okay, so I talked about the hardwired connectome yesterday, the monoaminergic neuropeptidergic connectome today, but so what we focus in the lab is on the electrical synapse connectome. So why so? Because if you look at that what are the electrical synapses? So most of the time when we study neuroscience in our school textbooks, like 90% of our attention goes to the chemical synapses. We call them synapses, and these are like kind of like left as a footnote. We call it a stepchild in the synaptic biology, but they're very important because if you look at the vertebrate brain development very earlier on, when the brain is still developing, you have a huge number of electrical synapses, and that is sometimes required for chemical synapse formation. So if you don't have the earlier electrical synapses, you don't have proper chemical synapse formation. They also are very important for left right asymmetry. This is not only in humans but also in C. elegans. So your left hemisphere is asymmetric to your right hemisphere in many different aspects, and these asymmetries in C. elegans and in higher vertebrates also, they are somewhat governed by these electrical synapses. The adult neurogenesis, like lot of memory formation and lot of behavior, neurodegeneration. So after neuronal injury, neurons like reprojects, axon gets severed, dendrites get severed, like now they need to regrow and connect again. So in those cases also electrical synapses come into play. Now are they important? Yes, because there are many people, you can see that they have hereditary hearing loss. I will give you one example. So the hereditary hearing loss, the 50% of the cases where you can trace the genetic mutation, 50% of them are can be traced to a single gene that causes makes electrical synapses. The other 50% we don't know, maybe they are electrical synapse associated genes. So now if you go for a genetic screen in the in Western countries, like where for a fetus or before having a baby, they do screen for these electrical synapse components because they are so prevalent. So your heart is these cardiomyocytes, if you put them in a culture, separate cells, they will all beat, they will all show synchronous beating, not all beats beating, but your heart, it needs to, all the cells need to beat synchronously, otherwise they cannot pump blood. So this synchronous beating is achieved by electrical coupling, and that is all because of electrical synapses. So they are very important. If you look at every single outside of the brain, they are also like present in every single organ, and their mutation is associated with disease that affects every single organ. So what are they? They are like multimeric hemichannels. What I mean that is a fancy term, but what I mean that there is a half barrel that are made by different proteins. So there are different proteins that oligomers to make a half barrel that is contributed by one cell, and when it docks against another half barrel from the other side, it gives you an entire channel. So that allows the ions to go from one side to another. One of the confusion that we have in the field is, most of the time when we look at the neuroscience, we say that electrical synapses are bidirectional. They are very fast and they are bidirectional. Fast, yes, that is a true nature of it. Bidirectional is not true, because the first ever electrical synapses that was discovered, it was unidirectional. Okay, so that's another problem, how this hardwired connectome, the anatomical connectome and the functional connectome can diverge, because in the hardwired connectome we always think the electrical synapses would be bidirectional, but in nature it doesn't. So if I give you an analogy, like suppose like if you draw a very simple network of A to A, B, C and D, if A is connected to B, B is connected to C and C is connected to D, by electrical synapses, if they are all bidirectional and all working simultaneously, like there is no particular regulation, once you stimulate A, everything will be stimulated, the whole circuit will be stimulated. But if you just go by this analogy, around like more than 250 neurons in C. elegans circuit, you can put it in a single circuit, which are like connected via, at least by different degree of separation, connected to by electrical synapses. But that is, then it is not possible, because if you connect the whole nervous system with electrical synapses and you expect all of them to fire together, where is this computation coming from? So that cannot be true. So there has to be some separation of electrical activity. So having an electrical synapses does not guarantee that it is going to fire at the same time, but what are the regulations? We do not know. Okay. So let us go into the details. Yeah.
And this is unidirectional because the junctions are voltage gated?
There could be many different ways that it can be bidirectional. One is the voltage gating. The other is like that if you think about like if you think about a barrel, just a simple barrel, how you can make it bidirectional? If you put like positive charge at the edge of the lip of the barrel, it will repel positive ions, okay, but it will allow negative ions, say chloride, but from this side it will allow sodium, because there is nothing hindering it. Okay. So by and different electrical synapse components have different sequences that puts different charge environment at the end, and also you can line these barrels with different charge moieties. That will also affect like what kind of ions it should pass, it should allow. If you take like sodium ion and potassium ion, there both of them are single charge positive ion, but the charge density is different, and having a different charge environment might interact with them. So some would be more permissive to sodium and not permissive to potassium, or vice versa. So that can also that is how you can also regulate. Yeah. And also like there are, I do not want to go into that details, but we are now finding like there are also proteins that forms another layer of complex structures on gap junction channels or electrical synapses that can also regulate whether which type of ions or which direction it can go and all that. Okay. So there are different mechanisms you can use to regulate which way and what type of ions which is going to flow. Okay. So so these are the structures. So there are hexameric or octameric channels that are coming from half channels that are coming from two sides and docking each other, and in principle this is a cartoon, but if you take an EM section it will also look like that. So individual channels we call it gap junctions, but in a electrical synapse sometimes you can have thousands and literally hundreds and thousands, like maybe 5,000 channels that are packed together and they are all simultaneously transmitting. So that together gives the electrical synapses. Okay. And what proteins make this electrical synapses? They are important because the two families of proteins, connexins in humans or vertebrates and annexins in invertebrates, that makes these channels. Okay. So one important thing is, like in every genome you look at, there are multiple of these connexins or annexins, meaning like zebrafish you have 40 connexins, in humans we have 21, and C. elegans there are 25. So why is it important? Because you can see that these gap junctions can be made out of different combinations of these proteins. So sometimes it is made out of like one single annexin, sometimes different combinations, and it is not that you can make any combinations. Some are more permissive, some of these combinations, some are not compatible with each other. So depending on which annexin is expressed in which cell, it can dictate whether the channel will form or not. Okay. And it is very conserved. You can take a human connexin, express it in C. elegans, it makes channels, it makes functional synapses. So the underlying biology that you can study in C. elegans, you can extrapolate it in humans in a very loose term. Okay. So this is how it looks like. So here there is a patch of electrical synapses in EM, and each of these dots are single channels. They are all together packed and like transmitting. And you can say that this is how it forms, like this is in another way of showing it. So half channels are coming from one side and they are getting together to form channel. Now what we know, so what was thought before, that there is a molecular lock and key type of mechanism that dictates where electrical synapses are going to form and where it is not going to form. So meaning that if a cell A and cell B can contribute compatible channel that can lock together, you will have gap junctions. In otherwise, if they are non compatible, you will not have gap junctions. So it is a very simple principle. Okay. So so now you can think about, like what he asked, like that what type of proteins, whether it is bidirectional, unidirectional. If you take that into account and this lock and key principle into account, you can actually try to explain two things that was actually proposed, that the expression of innexins or connexins guide two different things. One of the very important thing is synaptic specificity, meaning that what you have learned, like from the last I talked about in last sections, that in a bundle where all these neurons project, generally the neurites in a bundle. So in a bundle there are many neurons. So one particular neuron can have option to form synapses with multiple partners, but how it is choosing which neurons to form synapse and which is not going to be my partner neuron. So this is synaptic specificity. We can explain if the lock and key model is true, then if they have compatible innexins, they will form synapses, otherwise they will not form synapses. And the other thing is, if you have synapse, what type of property is going to be? It is going to have, is it going to be bidirectional, unidirectional, and many other different cases. So that can also be dictated by what innexins been firstly in the first place used to form these synapses. Okay. And let's, I mean, I'll try to show, like what are how what we think now. These are the assumptions we had when we started this project, and this is the molecularly distinct channels. You can also see there are different types of action. You can say that they can be like conducting, they can be silent, they can have like rectifying property, means going in one direction, bidirectional, strength of conductivity, voltage gradient. You can have all sorts of different things here. Okay. So now how to prove that this lock and key model is true or not, and how to explain what we see in the hardwired connectome. If you make a very simple ball and stick type of model, so if we know that okay, this neuron is connected to this neuron but not to this neuron, and if we know exactly what are the innexins that are present here, can we build this network out of these two informations? Okay, can we predict whether the hardwired connectome the way it is? Okay, so we have these lines already from the C. elegans connectome. So what we need to do, we need to put innexin expression information in that, and can we see that okay, the wherever we see orange, it can only connect to yellow but not to black. Okay, can we see this kind of principles, and can we explain the network? So yeah, so this is the C. elegans network if you filter out only the electrical connectome. So this is like where these lines are actually EM verified electrical synapses that has been observed in the C. elegans, and each of these nodes are different classes of neurons that are making these connections. Some of the lines you can see they are thick, some of them are thin, based on how big the synapse that has been observed in the EM. So this is a synaptic weight. Okay. So what are the different in, yes?
Previous slide just written that there's a condition an orange
yeah.
Connected to the but here I also noticed some self loops that the same entity is connected to.
Yes, it's actually interesting because I mean these self connections are between, they're not self because it's simplified network. It's if so these self ones are actually between left and right pair. So, most of the interneurons in C. elegans and in they are connected, left and right ones are connected by electrical synapses. What it does, it just gives you synchronous activity in the left hemisphere versus right hemisphere, but not all of them, because then otherwise everything will be similar. So, that is why you see some of them are interconnected. Yeah. So, every sorry. So, what is apparent here, that every neuron class makes electrical synapses, and the number can vary, because some of the hub neurons can make up to 30 electrical connections. Okay. So, how can we predict this? So, what I did in my postdoc, so I looked at the innexin expressions, so all of them in a single neuron level resolution. So, can we see where are these innexins are expressed, and can we find an innexin expression code of every neuron that can explain their connectivity. So, this is a innexin, names do not concentrate on the names. So, this innexin is expressed in almost all the neurons. These ones are most sparsely expressed, and here you can see that is expressed only in a single type of neuron. Okay, so, there are a great deal of variability. Now, if you take that all of it, like okay, so this one is two different isoforms of same innexin gene, and there you can see their expression is also variable. So, a single gene can have, like, probably you guys know that there are different splice isoforms or different isoforms that can come from same genes, but so there are same genes in two different forms that can also expressed in different places, but that gives you an added layer of variability. Okay, so what we found that there are many different genes, 10 of them are very broadly expressed, 4 of them are more restricted. So, out of 25 only 14 of them are expressed in the nervous system, but they are not exclusive in the nervous system, they are also expressed in the non-neuronal tissues, but in the nervous system there are only 14 of them, and you can see that their expression is quite variable in how many neurons classes it can be expressed. Okay. So, this is what it looks like if we put all these informations on top of this network. Okay. So, if you zoom into one part of this network, you can see that different neurons in this pie chart, their expression is showing like different innexins that are expressed. So, here this neuron is expressing 6, another one is expressing 4 in different colors. So, you get a very complex sort of combinatorial code of innexin expression. So, 80% of the neuronal classes, what we saw that they have unique combinatorial code of innexin expression. Okay. That, and other important thing is most of the neurons express multiple innexins, around 8. So, does it explain how is going to connect to its partner and where it is not going to be connected? Okay. So, now there is a loose first test that we did is like, okay, so some neurons, as I said, they make lot of connections, and some neurons make few connections, some neurons express lot of innexins. So, you can say that okay, the neurons that wants to make lot of connections need lot of innexins diversity, so that they can make like diverse connections, but we did not see any correlation between that. There is a very weak correlation. So, it does not so the number of innexins that are expressed in the neuron does not correlate to the number of partners it choose. So, what dictates it? So, I started with this hypothesis that can we explain the connectome based on this lock and key hypothesis. Okay. So, if the lock and key hypothesis is right, then if you see wherever you see the blue innexin, it will dock against the red one and you will see a connection. But what you see in the observed scenario is that many times you see compatible innexins that are expressed, but they do not form connections. Okay. Yeah.
Like sufficient to know whether certain innexins are expressed or not, or should we also care about how much one of it is expressed?
So, that is a good question. So, we do not have that information. So, I mean we have that information, but we do not know how to interpret it. How much is important, whether the The amount is also going to be a deciding factor, we do not know. So, here we just kept like whether it is expressed or not expressed, but yeah, it is a valid point. We did not want to like because we do not have that any knowledge at how it could affect the synapse formation. So, that is why we did not want that information. Okay. So, what could be the reason why we observe this situation where it does not match with the prediction? Okay. So, I will give you two specific examples. One is this circuit, again forget about the names. This neuron and it connects to this neuron with the electrical synapse, and with this neuron it connects with the chemical synapse, but all three of them has compatible innexins. So, what determines that this is going to be allowed, this is going to be allowed, but this is not going to be allowed. Okay. So, there must be a higher order of organizing principles. Again like look at this circuit. Okay. So, do not go by the names or the numbers, like, the concept is very simple. There is a green innexin that is expressed in this neuron, and we know that it connects to all these partners through electrical connection. So, there are valid electrical connections that are present there that has been observed by EM. So, it makes electrical connections, but we know that green one can function with the red one. Okay, they are compatible. So, what we know that for only for and the red ones are expressed in many of the partners, but what is surprising is that this green and red pair is used to make connections only for this synapses, but the other cases, although the compatible innexins are present, they are using something other innexins, they are not using this pair. So, what determines that? Okay. If you look at this is in the vertebrate retina. So, they are different. So, this is in the retinal layer, you have neurons in every different single layer. They all express this cone cells, this amacrine cells, they all have this connexin 36 expression. But what is interesting is that among different layers, within the same layer, there is electrical synapses using these connexins, but between two different layers, although they have the same connections, they do not use electrical synapses, they use chemical synapses. So, the two neurons that are present within this layer and present within this layer, they are making electrical synapses between themselves with the same protein, and between the two different layers they are making chemical synapses, although they have the proteins. Okay. So, what dictates this diversity? So, there are a lot of like computational work like that has been done with the electrical connectome as well, like from like with the David Hall's group and like Chaklovsky's group. This is the guy who is a fundamental like a key driving force behind the reanalysis of the electrical connectome, and also like this Barabashi's group, and some of the work has been done by Shakti and Shitabra also. So, that gives us some principles, the computational framework to look at these things, but we took a different approach. So, the questions that we asked is a very simple questions. All the neurons have different innexins, but they are using it very differently, that sometimes it makes synapses, sometimes it does not make synapses, sometimes it uses one innexin, sometimes it uses another innexin. So, the question that we asked, how a neuron utilizes is innexin or connexin expression repertoire. So, can we go to a single neuron and look at what are the genes that are expressed, where are these proteins are going, are they used for synapses or formation or not? If they are used for it, then yes, how it is done, and if it is not, then what is not done. So, we looked at this behavior, and that I showed here, that if you touch a worm it moves backwards. So, it is a mechanosensory behavior, and if you touch it in the back it goes forward, and we know this connection. We particularly focused on this mechanosensory neuron that is present in the back of the C. elegans body. So, it makes two defined electrical synapse zones, one is here, one is at the distal end of the neurite, and it gives us a good test bed scenario where it expresses 8 different connexins. So, we can say that okay, it makes connections with multiple different neurons, it has 8 innexins, how it is utilizing it. So, what we looked at, so firstly that was surprising, what was surprising to us is 5 of these innexins, although the mRNA level, like the transcription level is present, they do not, could not detect the protein, the proteins are not present. So, why? I have no idea, but probably it is because like the cell has some sort of safeguard mechanism for some rainy days. Okay. So, only 3 of these innexins are expressed. Now, you can expect that okay, now I have 3 different innexins and this neuron is making diverse connections. So, maybe it is going to use one for one connection, the other one for other connections. That is also not true, because all these innexins, you can see, you can label it in the genome, you can tag it and you can look at their expression, they are all are present in all the synapses. Okay. So, that is surprising, because you have 3 innexins, you have 8 first of all, you do not use all of them, you have 3 that you are used and then you are not even using it in separately. So, what does it mean? So, what we found, there is a cartoon representation that what we came, yeah.
In general, this is what looks.
In general, like I can't give you exact numbers, but in general, this is the thing. So,
no specificity.
no, not like we see. I'll tell you like that this organization that we see is not specific for PLM. It's also present in the higher vertebrates. I'll show you the data. So, so what we found that, as I mentioned, that electrical synapses are like clusters of different channels. And what we see that before it was thought that all these channels that are present within a cluster will be similar molecularly, one innexin or one connexin will make it. Now, what we see that there is a diversity of channels. So, this one innexin that is green, say is green, it makes channels that are all by itself, from one side is green, the other side is also green. Now, there are 2 other innexins that are yellow and red. They make like heteromeric, means like one side is red, one side is green, and these channels are arranged in a reciprocal manner, means like some of the channels are this way, some of the channels are in the other way within the same patch. So, what we can predict, because these these channels are similar from both sides, it will be bidirectional, and these channels that are like asymmetric in 2 sides, it has been shown to be unidirectional. So, from yellow to red. So, now you can see that now you put these 2 channels in a reciprocal manner. So, ultimately the resultant property of this whole patch would be bidirectional this channel and bidirectional combination of these ones. So, why we need this kind of complicated arrangement, like it would be much easier to have like one innexin making all these channels, there must be some logic to it, right. So, and Harish, like this type of organization is present in other circuits as well, other neurons as well. So, we call it like a heterochannel electrical synapses, that has not been shown before. Okay. But why do you need it? So, if you look at the chemical synapse counterpart, in the neurotransmitter receptor side, you see this kind of heterogeneity. Okay. So, this is a glutamatergic synapse. You have like ionotropic receptors that are like let us not go into the details, but these are like fast responding, these are metabotropic receptor, these are slow responding, but it lasts longer. And there are also differences between ionotropic receptors, that some are like gives you very sharp spike, some are give you little bit slower response, but little longer. So, there are also diversity in chemical synapse there, and these higher order diversities are very much required for to dictate how the synapse is going to respond, whether it is going to give you like just a short burst, or is going to give you like low dampened long time burst, and that will completely change how the network will function, because you, as I mentioned that in an intersection the cars are not coming all together. So, there is a time delay. So, this will allow you to create this time diversity, the separation at the time scale. So, is it like kind of similar in the electrical synapses? So, can you have this kind of, so are they doing something similar? But what is interesting is like that the first question that we ask, like that okay, are all these channels actually important? Okay, so now you can do, it's a very simple behavior that you can test in C. elegans. You can touch the worm and you can just see that how many times the worm is responding. So, in a wild type worm, if you take a eyelash and touch it, it always responds. So, it is a 100% response. But if you take, if there is a mutant that does not generate this sensory neurons, it never responds. So, you can mutate these genes individually and take out these channels and see whether do I need these channels, all of them, to get the response, or one is enough. Like what is the functional relation for having importance of having this together. So, now if you mutate one of them, there is no response, no defect, is perfectly responding as wild type. If you mutate each of them individually, there is no response. Okay. But once you combine, okay, so this is one thing. So, these two, this is a double mutant that takes out all of these green and red and yellow channels, but the green channels are still there. So, if you take out green, if you take out red, if you take out yellow or yellow red together, the worm still responds. But if you take out like any of these combinations, any of them together, that takes out all the channels, now the worm is completely not responding. Okay. So, that explains why you need these different types of channels together. Now, I will tell you like one other interesting thing, because so this synapse is very interesting, but it was kind of like mysterious, because this is the mechanosensory neuron. Now, it is activating your forward command interneuron, right, and it is also inhibiting your backward command interneuron. So, what it does when you stimulate this neuron, it inhibits the backward motion, because now it needs to go forward, because you are stimulating the worm like there is a threat at the back, and it is stimulating the forward movement. So, this is a chemical synapse, it is an inhibitory chemical synapse. This is an electrical synapse, okay, and that electrical synapse is that one that I was talking about. So, this is the single electrical synapse that connects this mechanosensory neuron to this command interneuron, and we did not, so before our work we did not know that there are 3 proteins that are present there, 3 different types of channels that are present there. We knew that only one type of channel is present there. People have mutated it and there is no response, no defect in the circuit. So, now you can see like for it puzzled the field for a long time, why you have a synapse that is sitting at such a crucial place in the circuit, but does not have any effect. If you mutate the protein, the worm still responds. So, now we see that it is not actually you can mutate one protein and get a response. There is a much more higher order structure of electrical synapses, and you have to take them all out to get a proper response. Okay, fine. Is it conserved? This higher order structure, is it only specific for C. elegans? So, what we did, we collaborated with Vatsala Thirumalai's lab. They work on zebrafish, and so this is a cartoon of a zebrafish larvae, and this is the cerebellum, and you can see this Purkinje neurons that yesterday I showed. They have very complicated but beautiful structure, and if when we looked at the single cell RNA-seq data, published data from Purkinje neurons, many different connexins are expressed here. So, we knew that this connexin 35B, which is little lowly expressed, it makes electrical synapses in that Purkinje neurons, that has been shown. So, what we took, like we made reporters of this highly expressed connexins, and we looked at whether there is any heterochannel type of this clustered together. So, what we see that these, so one of the connexins are co-localizing with the green and red, one type is co-localizing, the other type do not co-localize. So, means that one of them specifically is forming this heterochannel type of cluster in Purkinje neurons as well in zebrafish. So, what we see that is a fundamental principle of electrical synapse formation that we can discover in C. elegans with the innexins, and you can also extrapolate it in the zebrafish and see different types of families of protein, but the fundamental principles of electrical synapse organization remains the same, okay, which is really cool. But the other interesting thing is like so if I go back, the main interesting aspect of chemical synapse is you can change the ratio of these different types of receptors, they have different biophysical property. Now, in different cases, in case of our memory formation, there are two different types of receptors, AMPA and NMDA, you probably have heard the names or you will hear. There the ratios changes, and that is a crucial component for memory consolidation. Okay. Now, how can you change the composition here? If you think like that there is a green ball, and in a pool there is a green ball and some green balls and some red balls. If they are all tied together, green and red tied together, you cannot move them separately. You have to have them separate so that you can change the stoichiometry of green and red. Okay. So, here we know that you can take out some AMPA specifically or NMDA specifically, and you can change the stoichiometry, that will give you a complete different meaning of the same existing synapse. So, do we see similar things in heterochannel electrical synapses? Can we move the pieces together to make a different jigsaw? Okay. So, this we collaborated with Sandhya Kaushika's lab in TIFR, and here you can see in a live worm that this is the synaptic zone and this is the cell body of the neuron. We tagged one of these proteins in red, one of these proteins in green, and you can see that red and green are moving separately. The green is not moving that much, but red is like very dynamic. So, basically what you can see that within a same synapse you can move the pieces separately to form a heterochannel connection. Okay. So, this is shown in the cartoon that they are coming into different vesicles and you can change the stoichiometry. So, this is I am not going to go into the details. So, what we know now is like you can bring them with different motors. There are motors that are probably like kinesin, dyneins, there are different molecular motors. So, inside the cell there are microtubule tracks. So, and there are vesicles that are carried by these motors. There is a YouTube video that if you would like to see that, there is a I think it's called like life of a cell or something like that. It's a very good YouTube video. It explains how the cargo is moved from cell body to the synaptic vesicles are moving with motors. So, there are different motors that takes these cargoes and from the cell body to its destination, and we know that different vesicles are moving through different motors. We also know that they get degraded by different motors. So, they have like different way of putting it, different way of degrading it. So, you can change the stoichiometry. Yeah.
So why would they actually have so it would actually be bidirectional signal, then why would they?
I think that's a very good question. So, the short answer is we still do not know, meaning that in a behavioral assay, which is a much more crude assay, you can see whether animal is responding or not responding, because that is a very crude assay. What we need to do, there must be some minute biophysical differences, like the kinetics of the channel might be different. So, that is why both of them, although they are doing similar things, but there is a difference. That is what we think. We need to go into like the testing, like their actual signal transmission speed, and I think there we will see the differences. The other thing that I will say that, so I think another way to explain your question is why you need such a complicated way of arranging this bidirectional thing. Like, this is a simple way of doing bidirectional, there is a much more complicated way of doing bidirectional. So, I think what you can see that this is a simple way of communicating things. You have in one side you have the sensory neuron. When it is getting stimulated, you are also stimulating this command interneurons, right. But at the same time you need to manipulate the how much you are stimulating. If you are having a tonic activation of the downstream signaling pathway, you cannot quickly change direction, because now your forward circuit is very highly activated. So, it will take time to suppress it and then change the direction. So, meaning that suppose you are going in a you are moving forward, you are just walking, you can very quickly change direction. But suppose you are running at a full speed, it takes time, it will take time to change direction, because the inertia will be there. So, this is exactly the same way. If you stimulate your forward circuit too much, it will be very hard to change direction like instantaneously. So, you need to modulate how much you want to excite the downstream circuit. So, some of these bidirectional currents are actually flow back. So, they are actually kind of acting as a shunt, meaning that some of the current is passing from sensory neuron to the other side, but these channels, this is an hypothesis, I do not have proof, but what we think that this could be voltage gated, and once it reaches certain threshold, this reverse channel opens up and some of the current can come back. So, it just maintains the circuit activity, but does not allow it to go beyond that. So, this is exactly how any feedback circuit in the system works. So, there is a feedback mechanism. I think that is why you have this complicated arrangements, but yes, we need to do the much more quantitative assay to say exactly what you need it, why you need it.
Is the relative fraction conserved across the different series of the hetero?
We can't, I mean, in terms of microscopy, you can say that yes, they look similar, but in terms of exact numbers is very difficult to do unless you do a electron microscopy or that resolution, because each of these channels are 10 nanometer, okay, and the confocal or even very high super resolution microscopy does not give you resolution beyond say 100 nanometers. So, you can't go to the single particle level. So, that is the problem. Yeah. So, yes. So, what I talked about that there is a connectome, you also have this electrical connectome that gives you some of the innexine expression, and that somewhat explains the lock and key by lock and key model, but you have to have compatible innexine to form a channel, but having a compatible innexine does not guarantee you that it will always form a channel, because when we look at the network in this kind of way, what we do not consider that these nodes are actually 3-dimensional spaces, okay, 3-dimensional entities. So, this red one will only connect to this purple, another one will dock with the purple one when is present in this surface. So, suppose there is a so you are not transporting this red one to this surface, but you are putting it on this surface, it will never see if, that is partner. So, although there is so to give you a very crude analogy, so you can only get out of this door if it is present there. If you put a window there, you will not be able to get out. So, it is not necessary, is not sufficient to have a door in a room, the door needs to be placed in a certain way so that the communication can happen. Okay. So, here exactly the same way. So, this is a compatible innexine, it will form a channel, but you need to put it in the right place. If you are not putting it in the right place, it will not form a connection. Okay. So, so that's why, like, I didn't want to go into details, but we see that there are a lot of cell biological regulations that gives you where these proteins are going to go, whether it's going to be expressed, and how it is going to be, how long it's going to be present. So that dictates whether the synapse is going to be made there or not. Okay, the other thing is, like, that we can ask that is the electrical connectome plastic? I said like this NMDA, AMPA and all these things, but there is a plasticity. Is there any plasticity in electrical connectome? Okay. And if it is, what to what extent, and to what are the molecular mechanisms? So, simplest way of explaining plasticity based on this lock and key model, suppose this neuron is only connecting to this one of the partners because it has the compatible innexin and it does not have it. Now, if you express in a certain situation, this neuron starts expressing this blue innexin that is compatible, it will form synapses. So, you can get this plasticity based on innexin expression. So, if there is expression plasticity, that will give you more compatibility. Or you can also have, like, that you can change the molecular composition of these channels, like, this is like just by red and blue, and here you can add some green here, that will give you a different biophysical property, different way the synapse is like, functioning. This is unidirectional, this is bidirectional, you can also change it in that way.
You're just taking the protein subunits of this?
Yeah, you can make it like, different subunits.
Okay. So I mean, like, this is triggered by some stimulus? Or
Yes, I think so. We don't know about this subunits, that has not been observed, but you can actually do post translational modifications that has been observed. Okay, so and that changes that how the synapse will function. Okay, so this is an example of what happens in our eye. Okay, so you have our rod and cone cells that are heavily connected by electrical synapses, and how much the electrical connectivity will be is there, it determines how sensitive our eye is to light. So, if we during daylight, when there is like an ample light photons outside, we have less connectivity. So, you get like, but at night you get more connectivity. So, the signals can be amplified. So, every day with the circadian clock, the gene expression of these connexin genes, the expression of these connexin genes varies. So, it comes down during the day and it goes back again at night, and it happens every day in our with our eye, and these connections are so plastic. Okay. So, we again looked at the Dauer stage, which is like our favorite altered developmental stage. So, I showed you that they can do all sorts of different behaviors. So, it gives us indication that if an animal is doing so much different behavior, the nervous system must be undergoing lot of plasticity, lot of changes. So, it is a good system to look at whether the electrical connectome here is changing or not. So, now we know that this is in the adult stage, we have the atlas, we know the innexin gene expression, we have the connectome, but in Dauer, if we look at the innexin expression, if it changes, can we predict what the connectome would be, because we do not have the electrical connectome in Dauer. Okay, so, let us look at like how innexin expression changes. So, this is how it looks. So, we looked at the innexin expression. So, I looked at the innexin expression in non-Dauer situation, in Dauer situation, and then I overlapped with each other. So, yellow means it is expressed in both Dauer and non-Dauer, green means it only comes up in Dauer, red means it disappears in Dauer. So, now with a single gene you can see so much variability that a single gene can be upregulated in certain cells, at the same time can be downregulated from certain genes, certain cells. But if you look at the entire nervous system, so you can see that the same 3 neurons that I showed that expresses different genes, some of the genes are getting lost in Dauer, some of the genes are gaining in Dauer. Okay. So, does it mean that it gets new potential to form electrical synapses? So, what in the total nervous system context, 70% of the neuronal classes are showing up regulation or down regulation or one or the other genes. Okay, so in total 13 of them out of 14 are showing changes. So it's a enormous amount of plasticity that you can see in the nervous system, and nobody has looked at this kind of plasticity in the entire nervous system context. And now you can see that how much it can help to rewire the system. I don't want to give you data, but just want to mention it one thing. There is one single innexin gene that only comes up during the Dower state in one of the interneurons, that are the hub interneuron that receives signal from lot of sensory neuron. It only comes up in one single neuron in the Dower state specifically. When the animal comes out of Dower after feeding, this disappears, and this expression of this single gene connects this neuron to another sensory neuron, and it couples the left and right. Now, if you take this single gene out in Dower, if you stop this expression in Dower, there are specific aspects of worm behavior, chemotaxis behavior, locomotory behavior gets altered. Okay. So, now you can see that 13 different genes that are getting altered in 70 different 70% of the neuronal classes, how much it can add on to Dower behavior. Again, I will just go back to what I said yesterday, that the core principles of the connectome will remain the same. So, the worm will move in a certain way, they have the core principles will remain the same, otherwise the animal will die, but this extra layer of variability, these ones will give you stage specific behavior, stage specific locomotion, and this kind of certain advantage in survival and so on and so forth. So, this will give you the added layer of variability. Okay, so this is one level of plasticity. You can take that innexin expression changes and it rewires the circuit. Now, I left this here. So, there are different AMPA and NMDA that also changes. We have this similar sort of higher order architecture at the single synapse level. Does it also change? So, if you take this neuron, it makes two different synapses, both of them has different higher order structure, and when you take it in the Dower, when you look at in the Dower, this neuron, this synapses stays exactly the same how it was before Dower, but this synapse exactly in this synapse, so it loses these green channels only in this synapse. So, why? We still yet to figure out what is the significance of it, and if you feed the worm, it goes back in the regular developmental phase, and within few hours you start seeing the green ones are coming in and just going back to the regular structure. And we know exactly how there are differences in transport machineries that is required for these differences. So, now you can see that at the very higher level, at the circuit level, innexin expression gives you changes of innexin expression gives you rewiring, and even when you have a synapse, you can change the molecular property of each single synapse, that also give you a new dimension to the electrical connectivity. So, there are different ways of not only synapse formation, breakage, but also molecular plasticity of each of these synapses. So, altogether I think gives you a whole picture of how electrical synapse can function together. Okay. So, this type of synapse plasticity for electrical synapses, it has not been shown before. So, we are first one to report it, and let us see why we need it, this kind of things. There are a lot of more experiments that needs to be done. So, here I want to acknowledge I showed the works that are done by two graduate students, Atal and Sudhanand, and this is by have few more members, but this is kind of the lab, and the other peoples are doing different things to find out how nervous system. So, Akanksha is looking at how nervous system, so how we generate different behavior depending on the internal feeding state with the same circuit. then we are looking at many other aspects of electrical synapse formation and fundings and collaborators. And any question?
Questions?
Yeah.
So, the formation of this puncta or clusters of proteins forming electrical junctions.
These words you were
Yeah.
talking about. Yeah.
Is it a self-organized process, you think? Or are they collaborative?
Okay, so the problem is, like, that it has been thought like as a self-organizing process, because people have expressed them in heterologous systems like in cell culture, and there they self-organize. Okay, but in the neuron it's a very different context, because in cell culture those cells are not polar cells. Here neurons have polarity, their architecture is very different, their cell biology is little bit different. We do not know whether it is self organizing or not. Some data suggests that it is not self organizing, because what was previously thought that if you take a cell surface, these channels that are sticky channels that will go and float around in the surface randomly, and they will be like moving around with the membrane diffusion, and when they will find a cognate partner they will dock and form channels. So, that is a self organizing process. But now we know that they do not go all over the place. So, if they are going to form a synapse here, they get transported only near the cell body, and this position of the synapse are many cases defined, predefined by adherence junctions and other molecules. So, it is not is guided, but how much of it is self assembly and all that we still yet to know. Yeah, maybe that answers your question.
And they must also have a lifetime of these proteins.
Yes, so there it's a good question, because they have, some of them are very long lasting, some of them are very short, sometimes they have half life of less than couple of hours. So, what happens that new synapses are getting added. So, I think I will. So, here it is. So, if you see that these synapses in the patch, new half channels are coming from the periphery. So, the new half channel from this cell will come to the periphery, the other half channel will come to the periphery, then they will dock against each other and gets clustered. And so, the old channels are getting engulfed from the middle. So, there is always a convection going on excuse me, that brings the outer ones inside, and they are getting degraded from the middle. How fast the degradation is going to happen, that depends on, like, several factors, how the cell is responding and how the degradation machinery is working, so on and so forth. Yeah. So, yes, so the half life varies, and that is how it gets degraded. Yeah.
Gap junctions. Is there a cooperative effect? Like they interact and amplify opening of pores or something?
So, actually the data does not suggest that. So, some electrical measurement only predicts that there will be less than 25% of the channels are open at one point. Okay. So, but there are some interrelation, because now we can think that since we are now trying to we are finding that there are like hetero channel composition. So, some could be voltage gated, some may not be voltage gated. So, the voltage gated ones might have a cooperative opening, because the other ones will open first, that will reduce, and so after a certain threshold the voltage gated ones will, like, open at a certain point. So, that is how you can distinguish between low threshold signal versus high threshold signal. That is one way to do it. Yeah, the other way to do it is like not a cooperativity between electrical synapses, but in many cases electrical synapses and chemical synapses are present together very in close proximity, and activation of one can lead to opening of the another one. So, there is a cooperative effect between two as well. Yeah, that answers your question. Yeah. Yeah.
In the tower state, you said that there will be specific index expressed, which is not the non-tower state, right?
Yes.
So when they return back to non-tower state, how do they vanish in general?
So, they're regulated at the transcription level. So, we don't know about all of them, but for certain innexins we have looked at it. They are like in the dour state, which is like very starved condition, like there's less food, and generally food and insulin signaling goes hand in hand. So, if you feed them, the insulin signaling gets high and that represses their gene expression. Okay, and some of them gets down regulated. They also have like they are getting up regulated by insulin signaling, but we know only for handful of cases. I cannot say that we know 100% how much how it is regulated in all the neurons. Well, that's the simplest answer. I can give you a more complicated answer, but that's the simplest answer. That gives you the temporal specificity. Yeah. Okay, if there is no other question, then thank you guys.
So let's thank Abhishek for
It's like